So today's lecture will be focusing on study design and research methods and how they apply to skeletal muscle metabolism and some of the papers that we'll be covering in this class. A couple of announcements before we get started. Lecture captures have been posted to Sakai, so you should be able to follow a link on the sidebar termed lecture captures, and that should redirect you to YouTube where you'll be able to find all of the content for this course. Friendly reminder that presentations start next Thursday, so for the people presenting on carbohydrate metabolism, that's you. And the papers for the presentation are due midnight the day of your presentation. Also, the content in this course, you guys probably got a glimpse of it on Monday. It does move quickly. If anything doesn't make sense, please come talk to me. The sooner the better, just so that we can make sure you're on the right track before the information in this course starts to compound on itself and you feel like you're kind of left behind in the course material. Lastly, reminder that case study number 1 is due on Friday, February 7th. It's a couple weeks away, but it's time to start thinking about the papers you're going to use for this assignment. So we'll start off by talking about some types of studies. First and foremost, this all revolves around Latin terminology. So Latin is a highly descriptive language that is used in the sciences for centuries. Latin is this universal language where if I said anywhere in the world, if I was talking about something like a hemispherectomy, I could say that in Canada, the United States, Sweden, Brazil, wherever, because Latin is this universal language that we all understand, everyone would know that I'm talking about removing half of the brain in this procedure. So when we talk about types of studies within science, we use Latin terminology. So first and foremost, in vivo, meaning within the living. So what we're doing here is determining the effects of an intervention on whole living organisms. This is what most of the studies are going to be in this class, and it's usually referring to either human or rodent work. An example would be determining the effect of a high-fat diet versus a control or a normal diet on insulin-stimulated glucose uptake. Or we could even think about this from the context of ergogenic aids, where we think about the effects of beetroot juice supplementation on time trial performance. Next, we'll be thinking about ex vivo, so outside of the living or out of the living. What we do here is we can remove the tissue from an organism and expose it to an external environment that mimics its natural conditions. So we can take a muscle sample, put it in a petri dish that contains all of the same constituents of a normal muscle environment, and we can manipulate it from there. For example, in that normal muscle environment, we might add a high-fat solution, and we can actually look at fat transport, even though this muscle has been removed from the living animal. Another example you might have seen before is if we take a muscle sample tendon to tendon, we can actually mount that muscle to a force transducer, apply electrical stimulation, and look at how the muscle contracts or the contractile properties of that skeletal muscle, as opposed to having an exercising animal. This is a more controlled condition. Lastly, talking about in vitro studies, meaning within the glass. This is something we will see less of in this course, if at all. And really we're talking about experiments performed in a controlled environment outside of a living organism, meaning we're thinking about cell cultures in test tubes or petri dishes. An example of this as it relates to skeletal muscle metabolism may be the idea of culturing myotubes or muscle cells, and then looking at the signaling cascades associated and how we can manipulate that with different interventions. Thinking now into the idea of rodent work. So the things we need to consider are the types of rodents that we use for research. The main ones that you'll see, so the main types of rats are Weister rats or Sprague dollies. On the side of mice, we'll see something called a C57BL6 mouse. And basically what happens is that all of these rodent strains, within that strain, they are genetically identical. The benefit that this serves is that it ensures that the intervention we're using is unaffected by genetic differences or variability that may exist due to genetic differences. And really, big picture, it minimizes the inter-individual differences we could see in the response, meaning if an intervention works, it's likely to work the same in all of these animals. Next, it's important to consider ovarectomized rats or OVX rats. These are female rats with their ovaries surgically removed. Basically, this represents a state of menopause, and we'll see some of this mouse model or rat model in the sex differences section as well as potentially the aging section. By removing the ovaries, we're removing the ability to produce female sex hormones, meaning that we can study the effects with or without female sex hormones on physiology and metabolism. Big picture, rodents are important research subjects because we're able to manipulate a lot of their genetic traits or manipulate a lot of their physiological, metabolic, or even bigger than that, their phenotypic traits in order to answer the questions that we might have. Next, we need to consider knockout mice. So I mentioned in class that we can only have knockout mice, we can't have knockout rats. What a knockout model is, it's genetically modified in which the researchers can either inactivate an existing gene or a specific protein. What they do is they replace the gene with a non-functional piece of DNA, or they disrupt the gene of interest, making it non-functional. What we can do with this is determine the influence of a particular gene or protein on physiology or metabolism. So an example of this would be an estrogen receptor alpha knockout mouse, if we're interested in seeing how this estrogen receptor influences mouse physiology or metabolism. Next, we can have a different version of mouse models where there is overexpression or a knockdown of a particular gene or protein of interest. In the case of overexpression, we're thinking about increasing the expression of a particular gene, protein of interest, whereas in the knockdown model, we're thinking about decreasing the expression of a particular gene or protein of interest. This is more realistic than a knockout model because we're essentially upregulating or downregulating that gene of interest. An example of this would be in relation to Monday's lecture, thinking about increasing the expression of GLUT4 or increasing GLUT4 protein content in a rodent model. We could then see how that influences insulin or contraction-mediated glucose uptake. And on the other hand, we could have a knockdown model where we decrease GLUT4 gene expression and protein content and see how that influences skeletal muscle contraction as well as insulin-mediated glucose uptake. Another thing to think about is that sometimes the knockout models of the rat are not viable, so they cannot survive if we take out an entire gene or protein, meaning that they may be viable if it's simply overexpressed or underexpressed. So that's all I really wanted to talk about on the rodent side of things. Now we'll move into interventions that are typically seen in humans, and then kind of take that to the next level on once we have a muscle sample, either from a rodent or human, how do we analyze that sample to investigate skeletal muscle metabolism further? So why do we need to define our cohort? Really here we're thinking about who does this research apply to? Are they healthy? Are they overweight, obese, diabetic? Are they young? Are they old? Males, females, trained, sedentary, recreationally active, or elite athletes? By defining our cohort, we're typically talking about reporting measures such as height, weight, sex, or age, but we can actually go beyond these metrics and go further and think about disease states, body composition, or even aerobic fitness status or specific training styles. So really, the reason for this is that the findings in one cohort are not necessarily going to apply to the next. So if we have an intervention that works in overweight individuals, this may not be the case for healthy individuals. And this can all be linked to anthropometric, metabolic, or even physiological differences between these different groups of people. Another key thing about defining your cohort is that it allows future researchers to replicate your findings or attempt to replicate your findings in the same or a similar cohort. So the first thing I want to talk about is the maximal oxygen uptake test or the VO2 max test. This test is used to assess aerobic fitness status, so we're thinking about defining a group of people as either sedentary, recreationally active, trained, or elite. What this VO2 max test is an incremental exercise test that typically lasts about 10 to 15 minutes, and it's performed until volitional exhaustion, meaning that the participant runs on a treadmill or rides a stationary bike until they can no longer exercise or they feel like they're absolutely fatigued. What we do is we measure oxygen uptake and carbon dioxide production during this test, and we express their aerobic fitness status as their VO2, so their amount of oxygen consumed. We can express this in absolute terms, or we can express it in relative terms. If we're expressing it in absolute terms, we're talking about VO2 in liters per minute. If we're talking about relative terms, we're talking about milliliters of oxygen per kg per minute. The reason why we would express this data in relative terms is so we can compare the findings of one individual to another, regardless of differences in height, body mass, gender, things that may contribute to differences in a VO2 max test performance. For example, my oxygen consumption as someone who is of shorter stature and smaller body mass would be a lot smaller in absolute terms than someone who is 220 pounds and 6'2". So by expressing it per kilogram body mass, it's kind of accounting for these differences. So an average male who is recreationally active would be expected to have a VO2 of about 45 to 55 milliliters of oxygen per kg per minute, and an average female who is recreationally active would have a VO2 of 35 to 45 milliliters of oxygen per kg per minute. Now I want to talk about body composition. So determination of fat mass and fat-free mass in an individual's body, that is exactly what body composition does, where fat mass accounts for adipose tissue, and fat-free mass is everything else, whether that is muscles, bones, organs, blood, other fluids in the body. Importantly here, we're talking about the fact that two people could have the exact same height and weight, so they could be 5'11" and they could both weigh 200 pounds, but the way that their body composition is made up of fat mass and fat-free mass could be very, very different. So we need to use these measurement tools to determine both fat-free mass and fat mass in these individuals, as opposed to body mass index, which does not account for these factors. So now we'll talk about a couple gold standard methods for determining body composition. First and foremost, SKINFOLD calipers. So what we're doing here is measuring skin fold thickness at 7 sites typically, whether that's the triceps, the pec, shoulder blade, a number of regions on the torso, as well as the thigh. What we do is we add up our skin fold measures in millimeters and put this into an equation that can then spit out a body fat percentage for us. As discussed in class, some of the advantages of this method is that it's accessible, it's cheap, it's easy to perform relatively, and the disadvantage that we discussed was variability with regards to who is performing the test, and we need the same person to be performing that test from session to session in order to maximize the accuracy of our results. Next is DEXA, or dual energy X-ray absorptometry. Basically what this is doing is using 2 low-dose X-rays that are absorbed differently by bones and soft tissues. By these differences in absorption, we can actually determine the mass of the bones and the mass of the soft tissues, and then further look at those soft tissues in terms of fat mass and fat-free mass, and determine body composition from there. An advantage of this method is that the scan only takes about 3 to 12 minutes, so it's pretty quick. But a disadvantage would be the small amount of radiation that you're exposed to from the X-ray measure. Lastly, thinking about BOD-POD, so this measurement device is relatively new, and it's based on air displacement, meaning that we can take a baseline measurement with nothing in the BOD-POD chamber. What it does is it measures the volume of air in that chamber. Next, we can have a participant go into that BOD-POD And we can actually measure the volume of air that is displaced from the empty pod to when the participant came into it. And that volume of displacement is then used alongside the participant's body mass to calculate their density. Density is just mass divided by volume. And we know, based on physics principles, that fat mass and fat-free mass have different densities, which we can actually go on to use for body composition measurements. Again, an advantage of this is that it's minimally invasive, pretty quick. Limitation is that it's very expensive to have. And if you're claustrophobic, it's not really a great experience. Now I want to take some time to talk about some whole body measures and what that can do in terms of our understanding of skeletal muscle metabolism. So when we take whole body measures, what we're doing is actually gathering an understanding regarding accumulation and clearance of substances of interest. This can either be with regards to optimal metabolic function for health, or this can be with regards to even substrate utilization for exercise. So we mentioned this on Monday, but I figured I'd go over it again since it's fairly important and something you guys might come across in your first case study. So a glucose tolerance test, what we're doing is providing a bolus of glucose and tracking blood glucose over time in small increments, meaning could be every 15 minutes or every 30 minutes. In humans, we ingest a beverage called Trutol, which delivers 75 grams of glucose. In rodents, we can either have an oral glucose gavage, or we can deliver the glucose via intraperitoneal injection at 2 grams per kg body weight. And as we mentioned before, the idea that the oral gavage has this incretin response, so this hormone that stimulates insulin release, maybe giving us the idea that the glucose tolerance test results or our ability to clear glucose may differ when we have an oral gavage compared to an intraperitoneal injection. Big picture though, what this test is doing is looking at our body's ability to use insulin to take up glucose into those skeletal muscles. What the data actually looks like for a glucose tolerance test is we're measuring that area under the curve. So calculating the area under the curve in this case for our low-fat diet in the black dots, or in comparison to our high-fat diet with the white circles. And what we see is that the area under the curve is significantly greater for that high-fat diet compared to the low-fat diet. In this case, what this means is that there's higher circulating blood glucose in our high-fat diet animals following that glucose challenge, meaning there's an inability to clear the glucose load. In humans, what we would expect is that blood glucose concentration should return to baseline within 2 hours of consuming that Trutol or that oral glucose tolerance test drink. However, what we don't know about following a glucose tolerance test is how much insulin was actually used to clear the glucose load. So if we take that low-fat diet, black dots, for example, if we had 2 animals with the exact same response, we wouldn't be sure how much insulin was actually used to clear the glucose. So in one animal, it could be a smaller amount of insulin, so they're more insulin sensitive, whereas in another animal, it could take a large dose of insulin in order to clear the glucose to the same level, meaning they're probably more insulin resistant. On the other side of the equation, we can think about an insulin tolerance test. So this will actually tell us about insulin's action at the level of the skeletal muscle. So what we do is deliver a standard bolus of insulin, either intravenously or intraperitoneally. Remember, we can't deliver insulin orally because it's broken down during digestion and would not be able to elicit its effects on the target tissues, in this case, skeletal muscle. Insulin tolerance tests are performed in a fasted state. And what we actually do is, after delivering this bolus of insulin, again, we track our blood glucose levels over time, whether that's in 15 or 30-minute increments. And really the goal here is to induce hypoglycemia, meaning if we're in a fasted state, there's no food circulating, no insulin levels spikes or anything like that. But once we give that insulin bolus, there is that insulin spike, and its job is to promote glucose uptake into the skeletal muscle. If we take up more glucose into the skeletal muscle, we should be dropping those blood glucose levels to hypoglycemic levels. When we look at the results from the insulin tolerance test, what we actually see, same idea, we're measuring that area under the curve. So if we take our example of this low-fat diet versus this high-fat diet group, what we see is that the area under the curve for the high-fat diet group is, again, higher compared to the area under the curve for the low-fat diet. In this case, in response to an insulin bolus, what we're seeing is that in the high-fat diet group, there's higher circulating blood glucose, representing an inability to clear the glucose load via insulin, whereas the low-fat diet is able to clear that clear blood glucose from the circulation into skeletal muscle, inducing that state of hypoglycemia. What we can see here is that because we've delivered insulin directly, we can actually draw conclusions about the different rodents' insulin responsiveness. So we can say in this case that the high-fat diet group, because in response to insulin, they move less glucose into the muscle, they are insulin resistant. Whereas our low-fat diet group, because they had this hypoglycemic response, meaning insulin triggered glucose uptake into skeletal muscle, they are insulin sensitive. Another methodology we can use for the measurement of insulin resistance or insulin sensitivity is the hyperinsulinemic-euglycemic clamp. What this does, or what we do to administer this test, is the insertion of 2 catheters in the arm in a human. One syringe is going to continuously deliver an acutely elevated level of insulin. So hyperinsulinemic, meaning above normal, hyper being above normal. So a slightly above normal level of insulin. The other syringe is going to continuously deliver glucose in order to maintain basal blood glucose concentrations. So euglycemic means maintenance of normal blood glucose, which is typically around 5 to 6 millimole. This is considered the gold standard for determining insulin action in vivo, whether that is in rodents or in humans. And what I want to remind you guys here is that if we're continuously delivering a slightly elevated level of insulin, this is going to promote glucose uptake into our skeletal muscle in a healthy individual, meaning that we're going to have to continually infuse greater rates of glucose in order to maintain that blood glucose concentration. Whereas if someone was insulin resistant, the blood glucose concentration or the rate of infusion would be slower because of an impaired response to that insulin challenge. We see this in the data here, where what we're actually measuring is the glucose infusion rate, or the rate of glucose infusion. Basically, higher glucose infusion rates means that you're able to use insulin to clear blood glucose. And we can see that control individuals or healthy individuals have a higher glucose infusion rate compared to the high-fat diet here, who would be insulin resistant. So with this slightly elevated level of insulin, they're less able to clear the blood glucose, meaning they need to replace less of it, so they need less or lower glucose infusion rates in order to maintain a blood glucose of 5 to 6 millimole. Although this is considered the gold standard, something maybe we should consider again is this incretin response. So since we're delivering this hyperinsulinemic euglycemic clamp directly to the circulation, we're skipping that insulin response that would be mediated by incretins at the small intestines. So again, just something to consider in terms of methodologies. Maybe is this really the gold standard, or does this not 100% reflect what would be happening in the body? Now I want to talk about labeled tracers. So this is a very cool method in which we're able to deliver small doses of radioactive tracer, essentially containing chemical compounds where one or more of the atoms have been replaced with a radionuclide, where a radionuclide is an atom with excess nuclear energy, whether that is hydrogen, carbon, nitrogen, or phosphorus. Those are the key ones that you'll probably see in the scientific literature. And basically what we can do is, because there's a radioactive tag on these different chemical compounds, is that we can trace the distribution of that radioactive tag throughout the body. So for example, you'll see a 13N radioactive tag for protein metabolism, whereas you might see a 14C radioactive tag if we're looking at 14C palmitate for lipid metabolism. A couple of examples of where this has been used in real life. In Europe, they're allowed to do a lot more crazy science over there, and recently, one researcher actually fed a cow with radio-labeled grain. This cow ate so much of the grain that it actually produced radio-labeled milk, and they were able to use that radio-labeled milk to use it as a supplement in humans to track protein incorporation into skeletal muscle tissues as a protein metabolism as well as muscle hypertrophy study. Kind of on a similar note, heavy water. So what heavy water is that it's called deuterated water, or DTO, or 2H2O. Deuterated water occurs naturally. It's 1 in every 3,200 water molecules is one deuterated water. Basically what this is that normal H2O, or normal water, contains hydrogen ions with the 1H isotope. So that's the normal isotope of hydrogen. But water that is deuterated contains hydrogen with the 2H isotope. The 2H isotope is just different because it has one more neutron than the 1H isotope. And because of this, it has different physical and chemical properties than normal water. So how do we actually use this in science? We can actually use it to study protein metabolism in the sense that deuterated water is actually something that can be isolated, made into a beverage, and can be ingested by humans or injected intraperitoneally in rodents. And what we know is that these amino acids, or this 2H group, is actually donated to amino acids, which are building blocks for proteins, meaning we can look at how this 2H group, we can trace it to see how it's incorporated into skeletal muscle. So as amino acids labeled with that 2H isotopes, we can actually search out and find that 2H group because it has a different chemical and physical property than normal hydrogen found in water. And what we'll see in the protein metabolism side of things is that we can actually look at this deuterated water as a means to investigate protein synthesis rates, or some other terminology we'll see is something called a fractional synthetic rate. So this methodology is a little bit different than the other whole body measures mentioned before, but something we'll see in the ergogenic section of this course is functional magnetic resonance imaging, or fMRI. What this does is it measures brain activity by detecting changes associated with blood flow. So there's kind of a rule out there that when an area of the brain is active, it receives more blood flow, meaning that we can actually trace these changes in blood flow to look and visualize which areas of the brain are active. This is going to be important for assessing cognitive function in the sense of which areas of the brain are associated with memory, learning, attention, decision-making, those kinds of things. Where this will come into play is with an ergogenic called carbohydrate mouth rinsing, which actually stimulates reward centers in the brain, which can be visualized and may have a function in providing an ergogenic effect. So now we'll talk about measuring metabolites. So what I want to talk about in terms of measuring metabolites is how we can take a sample from the body and effectively use it to draw conclusions about skeletal muscle metabolism. So when we're measuring metabolites in humans, what we're doing is effectively determining fuel stores or the mobilization of those fuel stores as they pertain to different scenarios such as exercise. So we can take these measurements of muscle metabolites before exercise, after exercise, or even during. So first and foremost, blood sampling. So blood samples are typically taken from the antecubital vein in the arm into a collection tube. The blood is centrifuged, which is termed fractionation of whole blood. And really what's happening after that is we're separating the blood into its layers by mass. So first would be the plasma, which is the top layer. Second would be the buffy coat, which contains leukocytes and platelets, so we're thinking about immune response. And the bottom layer would be our erythrocytes, or our red blood cells. A lot of the proteins of interest for metabolism will be found in the plasma layer of the blood, and that's typically the section that we will collect for measurement. What I want to point out is that different tubes or different types of blood collection tubes are used for different blood measurements. And you can tell the difference between them because the tops of the tubes are different colors. Basically, each colored coat has either different anticoagulants in it, so we're preventing clotting, or different coagulants in it means we're promoting clotting. And it's going to depend on the measurement that you're trying to make, in the sense that serum refers to the plasma portion of the blood after mixing with coagulants or promoting that clotting effect. And plasma is the plasma portion of the blood when no clotting is occurring. So we have anticoagulants eliciting this effect. Some terminology you guys might run into with the research papers at hand would be things like an EDTA-coated tube, lithium heparin, sodium citrate, or even serum separator tubes. You don't need to know which tubes are used for which measurements. That's overkill, but just recognize that if you guys are to go on and research, you would have to think about these kinds of things. Another kind of blood collection measurement is the arteriovenous lines, or AV lines. What we're doing here, again, is inserting two flexible catheters in the arm to collect blood samples. Essentially, one is going to be in the artery, one is going to be in the vein surrounding skeletal muscle. In this case, what's happening then is that we can measure what's going into a muscle through the artery, and we can measure what's coming out on the other side in the vein. This can tell us about net uptake or net release of metabolites into skeletal muscle or from skeletal muscle. And by simultaneously determining blood flow rates, so how quickly blood is passing through those two sites, we can actually determine the rates of net uptake or the rates of release. An example of this would be the brachial artery. So brachial artery and brachial vein, we could take a sample from the brachial artery and a sample from the brachial vein to determine fatty acid uptake and release from skeletal muscle or from the biceps brachii. Another technique we can use is 31P magnetic resonance spectroscopy. This is a non-invasive tool for measuring the relative intracellular concentrations of phosphorus metabolites in skeletal muscle. So thinking about things like ATP, ADP, AMP, inorganic phosphate, phosphocreatine. What actually happens is that, similar to an MRI, we're using a magnet to attract electrons which have a negative charge, and we can actually compare them to set standards, whether that's phosphoric acid, or the fact that we know that different phosphorus-containing metabolites produce a certain spectrum on our scale here. So what we actually see is that we can quantify these different peaks. So we know a certain peak equals phosphocreatine, and we can quantify how many times that peak occurs to basically count the amount of phosphocreatine present in a sample, or how that might change prior to an exercise bout, so at rest, as well as following an exhaustive resistant exercise protocol, which is the figure on the top there. Another thing we can do, and probably one of the most major methodologies that we'll see, is skeletal muscle biopsies. So what we're doing here is obtaining a muscle sample, typically from the vastus lateralis in the quadriceps. What it is basically we're using something called the modified Bergstrom method. What this modified Bergstrom method is we apply a local anesthetic called lidocaine onto the muscle surface or onto the skin surface, which is basically a numbing solution or an anesthetic. We can then insert a biopsy needle into our vastus lateralis. The biopsy needle is pictured here, where it has basically an inner piece and an outer sheath. The outer portion of the needle is lifted and suction is applied, basically causing the muscle to fill that gap. Once the muscle has filled that gap, we can actually push down the outer portion again and basically cut that sample free and then analyze it. I highly recommend just looking up on YouTube how this process actually works if you're interested. It's pretty interesting and can provide a lot of valuable information in terms of analyzing our muscle sample. So what do we actually do with that muscle sample? So immediately after taking the muscle sample, it's frozen in liquid nitrogen. This is to preserve the muscle in the state it was in. So this is going to be extremely important for scenarios such as comparing rest and exercise. So in the case of exercise, we want to make sure that all the circulating metabolites that might be present in that muscle are actually collected and stored the way that they were at the end of that exercise bout. After we've taken our muscle sample, stored it away, and we're ready to analyze it, what we actually typically do is homogenize that muscle sample. So break it up so that all the fractions are equal in composition. In other words, we're going to essentially mash it up, break up the cell walls, as well as the mitochondrial membrane so that everything is present within our sample aliquots. Homogenization process can either be done through chemical or physical means. Chemical homogenization would typically occur through the provision of acids, whereas physical homogenization, we actually use ceramic beads to break up and physically destroy the cell membranes. This step is commonly used prior to determining mRNA or protein content. So the determination of mRNA or gene expression is through a process called real-time polymerase chain reaction, or RT-PCR. You guys don't need to know the nitty-gritties of how this exactly works. Just know that RT-PCR is a method for the quantification of mRNA species. Really what we're doing is determining gene expression. We can use a target mRNA that we're looking for, amplify it, and quantify how much is present. We can do this in real time. As for measuring protein content, we use a technique called Western blotting, in which we're able to detect specific proteins in a sample. The way that we can do this is through the use of gel electrophoresis, so the proteins are separated using an electrical current and by their molecular weight. What we actually do is transfer these results onto a membrane that can produce banding patterns for each protein of interest. The reason why we know certain proteins correspond to certain bands is that based on their molecular weights and comparison to standard proteins, we can determine which proteins are present in our sample. Another thing you'll notice is that we use a loading control as a quality control measure, meaning that what we're actually doing in a Western blot is looking at the staining or the banding patterns. So lighter pattern would mean that less of that protein is present, and a darker stain would mean that more of that protein is present. So using a loading control, we can actually show that I loaded the same amount of sample into each well, as opposed to overloading one, and that's why our protein content appears to be different. Typical loading controls include alpha-tubulin or GAPDH. The reason why this is termed a semi-quantitative method, again, is because we're quantifying how light or dark a band is. It's not giving us a specific number of proteins present in that sample. Another thing we can do with our muscle sample is determine enzyme activity. So this can either be determining maximum enzyme activity, or in some cases, enzyme activity can be expressed in physiologically relevant terms. It means that we might be able to determine enzyme activity with a concentration of the substrate that might actually be present in a physiological specimen. So what you'll typically see for enzyme activity assays is that they are expressed as specific activity. The reason for this is that we can actually look at the activity of the enzyme per milligram of total protein. In that case, we can actually standardize and compare across samples, meaning that we express it as micromoles per min per milligram. So micromoles of the product form. So remember that an enzyme takes a substrate, binds it, and creates a product. So we want the micromoles of product formed by the enzyme as a measure of how well it's functioning. We can express this relative to a minute so that the time is standardized, and we can express it per milligram of total protein so that differences in total protein content are not dictating the outcome of our results. For example, if we wanted to look at mitochondrial enzyme activity in trained versus untrained individuals, we know that trained individuals have greater mitochondrial content, so it wouldn't be fair to look at the activity of all of it together because the activity is going to be higher just strictly due to greater mitochondrial enzyme content. We want to express it per milligram of protein so we can compare the activity of one specific mitochondrial enzyme. In this case, we want to see if the mitochondrial enzymes, the activity is faster in trained individuals versus untrained individuals, despite the fact that we might have differences in protein content as a whole. Another thing we can do is look at glycogen content. So this is performed using a spectroscopy assay kit, meaning that what we actually do is look for color changes in our assay to dictate changes in or differences between our sample's glycogen content. So what we do is we take our muscle sample, we put it in each of these wells. We can actually then run this assay, which breaks down glycogen into glucose, and that glucose is oxidized, which causes a color change in our assay kit, meaning that if there was a greater amount of glycogen, more glucose would be broken down and more glucose would be oxidized, producing a greater color change. So what you might see is that at the beginning, our samples might be in this pink region, but if there's a lot of glycogen broken down to glucose and then oxidized, we might see this dramatic change to purple. Whereas if there was minimal glycogen content, what we might see is that a small amount of glycogen is broken down to glucose and a small amount of glucose is oxidized, meaning we only see this light purple color closer to the pink side. We can actually quantify this color change through something called absorbance spectroscopy, in which we use the absorbance of light as a final indicator of our glycogen content. Another thing we can do with our muscle samples is look at transporters. So whether that, in this case of this course, looking at glucose transporters or fat transporters that we'll talk about on Monday. We can actually measure total transporter content, which we discussed in class had some issues because now that we know insulin-stimulated and contraction-mediated glucose uptake, we need those GLUT4 molecules to be at the plasma membrane in order for them to have a functional role. So it's actually more important to determine GLUT4 content on the plasma membrane as opposed to total GLUT4 content, which could be intracellular, plasma membrane bound, we're not sure. So we can actually use a laboratory technique called differential centrifugation, which separates cellular components based on their mass, and we can actually isolate that plasma membrane to determine GLUT4 content specifically at the plasma membrane portion, which is more meaningful to us than possibly total GLUT4 content, which could mean intracellular as well as plasma membrane bound GLUT4. Next, I want to talk about immunohistochemistry. So this is taking very, very thin slices cross-sectionally of our tissue of interest, so 3 to 5 micrometers. And what we actually do is use a powerful microscopy-based technique to visualize specific cellular components. It's based on immunostaining, meaning that we can have antibodies that bind to specific cellular structures. So for example, in the green, blue, and red figure, we could have, this is for muscle fiber typing. What we can see is that specific muscle fiber types stain a certain color. So in this regard, we might have green being our type 1s, blue being our type 2A, and red being our type 2B. Because those specific antibodies are only binding to the specific muscle fiber types, they actually produce different colors, and we can then count and quantify the different muscle fiber types in our sample. On the top figure there, that's actually looking at mitochondrial morphology. So we have antibodies that can bind to the mitochondrial membrane. To visualize that outer mitochondrial membrane, we can actually look at discrepancies in mitochondrial structure and how that may contribute to mitochondrial dysfunction or disease. So on the note of mitochondria, I'd like to move into some mitochondrial measures. So mitochondrial DNA, so first and foremost, mitochondrial DNA, or mtDNA, is quantified using that RT-PCR that we talked about before. And it's often used as a marker of mitochondrial content, in the case that more mitochondrial DNA often means greater mitochondrial content. Decreased mitochondrial DNA, decreased mitochondrial content. So for example, a trained individual would have higher mtDNA versus an untrained or sedentary individual. Another term that can be used for mitochondrial DNA is mitochondrial copy number. Next is transmission electron microscopy. So this is used to determine the structure of mitochondria within a cell. What we actually do is we fire electrons at a specimen. Some of it reflects off and other parts pass through. If it reflects off, it creates a dark region. If it passes through, it creates a light region. So what we see is that through this light and dark regions, we actually create a structure, in this case, of a mitochondria. And it can also be used to determine mitochondrial dysfunction. So again, we can look at and think about the shape of the mitochondria, where elongated mitochondria are considered to be unhealthy or improperly functioning. As previously mentioned, we talked about differential centrifugation. This method is common in mitochondrial preparations and can be used for isolating mitochondria. What we're again doing is using centrifugation in order to separate the different components of a cell based on their mass. What's happening is the cell membrane is ruptured and releases the cellular components, meaning we're able to isolate the individual organelles of that cell. Again, used for isolating mitochondria or other cellular components. If we isolate the mitochondria, we're actually able to determine the content of specific mitochondrial complexes, or even determine the activity of those mitochondrial complexes. If you are someone who studies the mitochondria, a technique that you will run into is something called permeabilized fibers. So what we're doing is using skeletal muscle samples and applying something called saponin. Saponin is a soap-like toxin. What it does is that it foams when it comes in contact to water. And saponin is interesting because it only lyses the plasma membrane, but leaves the mitochondrial membranes intact, meaning that we're able to use this method in order to study mitochondrial respiration in an intact model. So what is mitochondrial respiration? Mitochondrial respiration is essentially movement through that electron transport chain and how that's used to form ATP. We can study mitochondrial respiration and the function of a number of these mitochondrial electron transport chain complexes by the provision of a number of substrates to each complex. So if you ever see a paper that says they applied pyruvate, glutamate, and malate, they're studying complex 1 of the electron transport chain, because those things can go on to produce NADH. NADH feeds complex 1, which we then know can go on to complex 2, 3, 4. If we're talking about the provision of succinate, we're talking about the production of FADH2, or feeding directly into complex 2. If we're talking about complex 4, we'll be delivering something called TMPD ascorbate. What this does is actually just provide electrons that can then feed ATP synthase. So the last thing I want to talk on the mitochondrial front is mitochondrial complex inhibitors. So if we want to inhibit complex 1, we can use something called rotenone. And if we actually use rotenone to inhibit complex 1 and deliver succinate, which again goes straight into complex 2, what we can actually do is determine mitochondrial function in the presence of rotenone and succinate, as well as mitochondrial complex function as a whole. And if we subtract our mitochondrial complex function as as a whole, and then we take away everything except complex 1, we can then deductively reason what complex 1 was contributing to that picture. Alternatively, we can inhibit complex 3 using something called anti-myosin A. If we are then basically minimizing flux through complexes 1 and 2, and this is also where we can look at mitochondrial reactive oxygen species production. Inhibition of complex 4 occurs through something called oligomycin. This is also used to measure something called leak respiration, which is basically not coupled to ATP synthesis. Lastly, we can have CCCP or FCCP, which is used to measure maximal mitochondrial respiration. So basically, how does oxygen move through the system maximally? So we can do that without the presence of CCCP or FCCP, and with the presence of this, and compare how the electron transport chain functions. So I want to end off on a little bit more of a casual note, thinking about measuring exercise performance. So first and foremost, aerobic time trial performance is measured as either a set distance, whether that's 5K kind of race, a set time, how much work can you do in 60 minutes, or how far can you run in 60 minutes, or a set amount of work. So if you want to standardize the protocol for everyone involved, we can do, for example, 4 kilojoules of work per kilogram body mass. Alternatively, we can have a time to exhaustion protocol in which you run until you can't run anymore. But when we consider aerobic time trial performance, what we need to think about is something called ecological validity, or how much does this apply to a real-world setting, where a 5K race would be a lot more ecologically valid than a time to exhaustion protocol, because you would never have a scenario, hopefully, where a human is tested on their ability to run to exhaustion. Other performance metrics that you guys will come across is in strength, so more on the protein side of things, repetitions to failure at a submaximal workload, or measurements of a 1 repetition maximum. In terms of stop and go sports, the yo-yo intermittent recovery test is very similar to the Loughborough shuttle test, and both of these are very much like a beep test in the sense you run from point A to point B and then have a rest. So in the yo-yo intermittent recovery test, you run 20 meters there, 20 meters back, recover until you're called on to do it again. The breaks get shorter as the test goes on, and it's supposed to mimic stop and go sports performance. So we think of soccer, where you might be running as fast as you can, taking a break, walking, running as fast as you can, taking a break, walking. That's the goal of these tests. Again, they have quite a bit of ecological validity because of that. Last, I want to talk about supplement considerations. So will the supplement actually work? So things we need to consider as scientists when we're thinking about studying ergogenics. How is it dosed? So is it an absolute dose where we're given 500 milligrams or 1,000 international units? Is it relative to body weight, so 3, 4, 5 milligrams per kilogram body mass? Usually it's dictated by how it's administered. So if it's commercially available, you and I are both going to use the same scoop. We're going to take the same bottle off the shelf. We're going to take 2 tablets of the same thing, regardless of how much you or I weigh. But if it's prescribed, it could be dosed relative to body mass, depending on how, the type of pharmaceutical. And again, this is not always the case because there are sports supplements that are administered as milligrams per kilogram body mass, such as caffeine, or there's even pharmaceuticals out there that are delivered in absolute doses too. But again, these are all important things to consider, and does it matter or does it make a difference if we deliver it as an absolute dose or if it's standardized to the weight of our participants? Next, we need to consider whether it accumulates in the blood. So most supplements or pharmaceuticals are administered orally. So what we can actually look at is something called the pharmacokinetic profile. So how quickly does it accumulate in the blood? We can track blood samples once an hour for a 24-hour period, for example, see when it peaks in the blood, see when it declines back to baseline levels. That would be our pharmacokinetic profile. Or, alternatively, one thing that would suck and would not be good for a supplement is that if it didn't accumulate in the blood at all, meaning that it's excreted out in the urine or it doesn't survive first pass metabolism. We can then look at things like a dose response. So if I give a small dose versus a medium dose versus a large dose, does that largest dose elicit the largest effect, or does it not work that way? Alternatively, we can consider acute versus chronic. So if I take this supplement or this drug every day for a year, is my response different at day 1, month 3, month 6, at 1 year? Does it accumulate over time? Or does it get cleared every single day and we start over on a 24-hour kind of period? Again, all things you want to consider when you're thinking about a supplement or a drug of interest. If it's going to be efficacious, it needs to survive first pass metabolism. It needs to get into the blood to have a chance to even get to our target tissue, which in this course is going to be skeletal muscle. So now thinking about if it's passed its first test of getting into the blood, does it get into our skeletal muscle? We can now determine this knowing the tools we have with a skeletal muscle biopsy. If we take our muscle sample, we can actually determine, look for the protein content of our main bioactive of interest and see whether it actually accumulates in the muscle. So the same kind of principles can apply here. We can have a dose response, we can have pharmacokinetic profiles. We want to see how that supplement changes in the muscle over the course of our supplementation or our drug ingestion period. With that, those are all important things to consider when we're considering supplements or pharmacological agents. On Monday, we're going to have another heavy lecture talking about lipid metabolism. And on Thursday, we will have our first round of presentations with 5 people talking about carbohydrate metabolism.